can you tell me a little bit about yourself and what motivated you for your involvement in or your creation of the company?
Okay, sure. So I'm a biomedical engineer by academic training with over 22 years of experience now creating and commercializing new medical devices and some other technologies along the way as well. I started out as an R&D engineer designing neurodiagnostic equipment and gradually moved into product management and more market-focused roles with increasing responsibility while working for a series of both early stage companies and also the global corporations that acquired them. And then back to the next early stage startup. And those experiences were with a mix of neurotechnology, imaging, some surgical navigation, and a bit of optics in terms of scope of the field. The scope of my work now includes a fairly broad range of medical technology, innovation, and commercialization activity. Topics including strategy, technology assessment, market analysis, health economics, requirements definition, product development. Let's see, there's intellectual property, regulatory affairs, business development, marketing, sales. You know, there's a lot of pieces to the puzzle, and I've been fortunate enough in my career to have exposure to most of them. Yeah, in my spare time, I'm also a mentor for startup entrepreneurs, and I'm an external reviewer and a judge for a few different technology funding programs and award programs. In terms of SWAN Cytologics, so when I was first introduced to SWAN, what really excited me was that I found that the clinical benefits that this project was offering, the technology maturity, and of course, the strength of the value proposition were all highly compelling. I was also very impressed with the founding team. So the founding team consisted first of a pathologist, Dr. Ken Pritzker, who's the former head of pathology and laboratory medicine at Mount Sinai Hospital here in Toronto. So he has many decades of experience and a very deep understanding of the clinical need from the pathology side. He's also been part of several other startups. So he's got a bit of a track record as a serial entrepreneur. So he together with an accomplished ultrasound physicist, that's Heikki Nieminen, who's the director of the Medical Ultrasonics Laboratory at Aalto University in Finland. And he's the one who designed this elegant solution in response to the clinical need that Ken had identified. And the two of them together with John Jordan, a business executive who helped them to create and run the company. He has experience building other companies from startup through to exit. So it was a pretty impressive group and an impressive project. And at the time, they were looking for someone to help with the commercial translation of their project. And you know, naturally, when they offered me the chance to get involved, it was just too good of an opportunity to pass up.
Now, what problems does your company address?
So at a high level, the problem that we're addressing, or the problems that we're addressing, are the inadequacy, inefficiency, and trauma that go along with needle biopsy procedures. So to give you just a little bit of background, needle biopsy is a very common diagnostic procedure. It's used extensively in cancer care, but also for other applications. And it's a crucial step for precision medicine therapies. And it's a procedure in which a needle is inserted into the target, the suspected tumor perhaps, to extract a sample of cells or tissue for analysis by a pathologist. And the pathologist will try to determine, for example, the presence, type, and characteristics of a cancer. There's two main types of needle biopsy today. Those are fine needle aspiration and core needle biopsy. And together, there's about 10 million of these procedures performed annually at a cost of about $10 billion. So fine needle aspiration came first. It was introduced, I think, back in the 1930s, and it uses a very thin needle to obtain a small sample of cells. But the challenge there is that the sample volume and quality are often inadequate for the purpose of taking the sample, and it doesn't allow for assessment of tissue structure because you're only collecting a sample of cells, not intact tissue. So eventually core needle biopsy was introduced. That came much later, although now it's actually used much more frequently than fine needle aspiration because it uses a much larger needle to collect a larger sample. So you get more material for analysis. However, that comes at a cost. The larger needle causes more trauma to the patient. The larger sample actually results in more complicated sample handling and sample processing in pathology. It's a more expensive procedure. And still, despite all these limitations or these changes, still the samples are frequently inadequate for a complete pathology assessment, either due to mechanical damage to the sample or degradation, or maybe blood contamination or some other factors. And I can give you, some peer-reviewed article references if you wanted to really go into detail around the different types of challenges and the different types of biopsy procedures and when and where those are experienced.
Okay.
But in any case, with these problems, so that's where Swan Glide was introduced. It's intended to solve these problems by collecting a better quality, easier to process sample than core needle biopsy, but at a lower cost, and while using the smaller sized, less invasive needle of fine needle aspiration.
Who specifically experiences the problem that you're addressing?
It's an interesting workflow I can take you through. I mean, really, the problems that I described really do affect all the stakeholders who are involved. So from the clinical perspective, the clinician, and there are several clinicians involved in this biopsy workflow, they're really not able to provide the quality of care that they strive to provide for their patients. So that's a frustration for them. From the patient perspective, they have to suffer through the increased physical and emotional trauma and pain, while also paying a higher cost, and there's greater inconvenience. And in the end, the outcomes are not as good as they could be. And then the economic stakeholders who are involved, and that can depend on which health system, which model you're talking about. But the economic stakeholder has to deal with the financial waste involved in these procedures. And also, this is really from a global perspective in this marketplace, it's a constraint on many of the dependent technologies that rely on good biopsy samples. So if we step through the process, here's what would happen. So a patient has a suspicious mass or lump. And the referring physician decides that they need to have a biopsy. And in almost all cases, when there's a suspicious mass or lump, the next step is a biopsy. It's rare that you'll proceed to treatment without collecting that biopsy and characterizing the type of cancer or confirming the presence of cancer before recommending a treatment. So the referring physician now has to make this choice between fine needle aspiration and core needle biopsy with the trade-offs that I just described earlier. Sometimes they'll do that by starting with fine needle aspiration and then switching to a core needle biopsy after the fine needle aspiration doesn't work. But as I said, it's much more common now to go straight to core needle biopsy. There's some types of biopsy, lymph node, for example, thyroid, for example, where fine needle aspiration is still maybe more common. But that's step one. So the referring physician has to make this difficult choice between the trade-offs of the two procedures. So next, the patient ends up with the physician who's going to perform the procedure, the biopsy. Maybe it's an interventional radiologist, maybe a surgeon. In some cases, it might be a pathologist. So now the physician has to either struggle to get a sufficient sample using fine needle aspiration, or they're using core needle biopsy. And they have extra work to do with patient pain management. So maybe some anesthetic delivery, also wound closure after the procedure, maybe having to stitch it up instead of just putting on a Band-Aid. And also there's a higher risk of adverse events, things like bleeding and other complications. So, and in either case, whether it's fine needle aspiration or core needle biopsy, they may have to perform some extra passes, target the sample aim for the target a couple extra times to get enough samples before they have good enough quality samples to send off for the pathologist. Now the pathologist has this challenging task of providing a meaningful diagnosis or treatment recommendations or recommendations for further diagnostic workup based on what might be an inadequate or compromised sample. So you end up with longer processing times while they're doing this, extra work for the pathologist. Although I will say that the pathologist will take, they'll stop at nothing short of heroic measures to extract as much information from these samples as possible, because certainly they don't want to put the patient through having to go through it again, and they'll do their very best at finding as much meaningful information as they can, but often they don't have conclusive results. They have things that are consistent with a particular finding or suggestive of or indicative of, as opposed to being able to make a absolute determination. So now the report from the pathologist goes back to the referring physician, and the referring physician now has to make a decision about diagnosis or treatment based on that imperfect or inconclusive information. And especially something like if you wanted to select a precision medicine therapy, you can't direct a patient towards precision medicine without having the right molecular diagnostics or other protein markers or things that have been identified before you can choose the right precision medicine therapy. So maybe they send their patient for another biopsy, or maybe they take what information they have and they go ahead. I actually spoke with someone recently who was telling me their father actually had several failed biopsy attempts, at which point his doctor said, okay, you know what? We're not going to keep doing this. Let's go ahead with the surgery. Let's go ahead and remove it. They went performed the surgery, removed the mass, the tumor, and turned out it was benign, and they could have spared the patient from this. That's the kind of thing that goes on. So the patient, right? Let's not forget about the patient. Obviously, the focus of this, the patient now has gone through this whole process. You have the trauma from the procedures, both emotional and physical. You have delays between initial entry into this workflow and finally reaching a diagnosis and being directed for treatment. And so those delays lead to worse outcomes. And of course, there's the extra costs and all the other inconvenience that goes along with this workflow. So from the economic perspective, if the procedures need to be repeated, if the information is not useful, there's all wasted money spent within the healthcare system, which is not acceptable. And that's just considering the direct costs. If you look at the indirect costs as well, it's an enormous problem, especially with things like the growth in precision medicine, where a significant number of new therapies being introduced are precision medicine therapies, but they can't work without a good biopsy sample. So this problem is going to get worse and worse as new advanced treatments come into the clinic. Yeah, and the other challenge that you get is because of these challenges in the workflow, biopsy is not routinely used for monitoring. I mean, how many patients would willingly subject themselves to routine biopsy during therapy, and without it being reliable enough, how many physicians would even ask their patient to do that? So things like response-guided therapy, which can be very beneficial in some cases, are really very underutilized. And that's actually part of the origin of SWAN, came from the founder's experience with a previous startup that was performing RNA analysis on samples to help guide treatment decisions. And they were having a lot of trouble getting good samples at efficiently and effectively getting those good samples. And it was while brainstorming about, well, what do we do to solve this problem, that they actually came up with the original ideas that led to Swan Glide and SWAN Cytologics.
Excellent. You've shared a story about somebody who had multiple biopsies and then ultimately it was benign. Can you share any kind of other example of how somebody experiences this?
It could be that procedures need to be repeated, which is cost, inconvenience, and pain. It could be that treatment decisions are made based on insufficient information, and you may find down the road that it wasn't the optimal therapy that was selected. I don't want to say they were given a bad therapy, but I talk about the problems, but let's be clear, the clinicians involved here are doing the very best job that they can with the best, to the best of their abilities with the tools they have. There's no one out there who's saying, yeah, it's whatever, it doesn't matter, what's the big deal? They are trying their hardest and they're doing a very good job. So I don't want to say patients are getting bad therapy, but they may not be directed towards the optimal therapy. In many cases, they're having core needle biopsies performed when a better fine needle aspirate or a better sampling with a tool that could get them a better sample, but with a smaller instrument, would subject them to much less pain, trauma, scarring, and of course the risk of adverse events that go with it, bleeding and the like.
Excellent. That's all good stuff. If you wouldn't mind, talk about your solution as if you're speaking to an eighth grade class, eighth grade science class.
The way we would explain it, so what Swan Glide does, it uses ultrasound energy. So ultrasound energy is high frequency sound waves. And what we do is we send these sound waves through a fine needle and into the target, the tumor that we want to sample from, or the mass that we want to sample from. And what these sound waves do is they cause some gentle vibrations of the needle tip. And that causes a little bit of motion in the needle tip, which helps to draw the material into the needle. And also, as those energy waves pass into the sample, they stir things up a little bit. They soften the target in a sense, and make it easier to pull tissue or cells up from that sample. So it increases the sample uptake. And also this acoustic energy, these sound waves, also help to reduce resistance to the needle while it's being inserted. Kind of like if you imagine someone, an Olympic diver, right? If they dive into the water nice and straight, they break through the surface tension very cleanly, and it's not a big, not a very large splash. So you can do that also if you break the surface tension with something like an acoustic wave before pushing your needle through, as opposed to the other approaches where what happens is you push the needle through the skin, and basically you stretch the skin until it ruptures, and so it causes a much larger wound.
What are the biggest challenges that you see out in the future?
Yeah. Well, adoption of new medical technologies is always a challenge. There's a lot of inertia in the healthcare community, and rightly so. I mean, we have to be very careful about what we introduce. There's an interesting paradox that if you want to introduce something new that's better, if it's better, that means it's different. But in the medical community, different is bad. And
Right.
so, by definition, if you close that loop, anytime you introduce something new in the medical field, it's bad. But in our case, we think we have a very compelling solution. And as I described earlier, all the stakeholders really have something to gain here. All the different clinicians, the patient, the economic, the payer or economic stakeholder. So it's really, it's a win-win-win. And we think that will really facilitate that adoption in the market. The other big challenge is I'll mention again, is that funding round we want to secure, because obviously without the money, it would be more challenging to accelerate our efforts and proceed with those next steps towards the milestones.
Anything else that you'd like to share with us that I haven't asked?
It's pretty thorough. I think we've covered it all. SWAN, in my opinion, my biased opinion, I think really checks off all the boxes. There's an excellent fit here between an identified clinical need and a technical solution that was designed specifically to address that need. A solution which also I think is very elegant in how it works. Keep in mind, right, this project, as I said earlier, is fundamentally driven by the clinical need that was identified by a clinician, and not that it's a technology that an engineer came up with that we're trying to push out into the market by any means available. The opportunity that we're looking at in this market, it's a large and growing opportunity, and that growth will accelerate. I think biopsy will be utilized more when Swan Glide makes biopsy a better tool, more effective, more efficient tool. And also with the growth of things like precision medicine and response-guided therapy, we'll see a lot of growth in this market space as well. I think we have a really strong founding team in place. That's definitely, something to highlight. Our initial validation results, we're not just talking about something in theory. We've shown data showing the improvement in sample yield, showing the improvement in quality of the samples, showing the reduction in trauma. These are all things that we've demonstrated, not just things that we're speculating about. Our IP position is very strong as far as giving us a competitive edge, a defensible barrier to competition. Our international PCT application comes with an initial search report that comes back. The examiner found that all of the claims in our PCT filing were novel and inventive, which is surprising but very exciting first response to a patent application. So we're off to a great start there. And on the financial side, it's a very attractive financial model, very attractive return on investment to our investors. And we'd love to have good people involved who see the opportunity not just from a financial perspective, but also for bringing a really important clinical advance for the sake of the patients, for the sake of the clinicians, for the sake of the healthcare systems. We've estimated there could be savings of as much as $2 billion a year to healthcare systems around the world by introducing Swan Glide and by migrating from core needle biopsy and fine needle aspiration to these Swan Glide procedures. And of course, at the same time, generating a pretty attractive revenue stream for Swan as well.
Great. Excellent. Thank you so much for your time today. I know you're busy and
Pleasure. Always happy to talk about my pet projects.